IB Bio, Plant Biology Part 8, Reproduction C, will focus on the seed, its structure, and the conditions under which the seed germinates. The essential idea is reproduction in flowering plants is influenced by the biotic and abiotic environment. Here's an outline of all the available movies for plant biology, for higher level students only. Use this outline to find the movie you need for review. This movie is focused here. Plants are composed of roots, stems, leaves, and flowers. Flowers are the reproductive organs of a plant. Meiosis occurs in the flower to produce both the male and female gamete. Pollination, then fertilization, together result in seeds being formed within the ovary of the flower, and then the ovary develops into a fleshy structure that surrounds the seeds within the fruit. The fleshy fruit facilitates seed dispersal away from the mother plant. Then seeds germinate when the conditions of temperature and water are suitable for growth. Angiospermophytes are the flowering plants, and the presence of a flower is the most recognizable characteristic of the angiosperms. Angiospermophytes have a protected seed. Coniferophytes have a seed that is less well protected than angiospermophytes. Mosses and ferns do not produce seeds. Seed structure and germination are the two concepts covered in this movie, the last of the plant biology series. This movie has its focus on the seed, which is composed of a diploid embryo, nutritious endosperm, and a protective seed coat. Let's look at the life cycle of a plant, starting with the flower. Meiosis to produce the female gametes occurs in the ovary. Meiosis for the female gamete occurs specifically within the ovule within the ovary. Meiosis to produce male gametes occurs in the anther. The male gamete is enclosed within a structure known as pollen. Pollen from one plant reaches the flower of another plant in a process known as pollination, which is gamete transfer. Once the pollen has landed on the stigma, sperm will move through a pollen tube to fertilize the egg that lies within the ovule. Fertilization is the union of sperm and egg to produce the diploid zygote. The diploid zygote develops into an embryo that's enclosed within the seed. The seed has nutritious endosperm and a seed coat. Now, the fleshy structure that develops around the seed comes from the ovary and serves to facilitate dispersal of the seed away from the mother plant. And then, under the right conditions, the seed germinates and grows into an adult plant, a reproductively mature plant that can flower. The sperm nucleus will reach the egg that is within the ovule within the larger ovary. Fertilization occurs in the ovule. The second nucleus will fertilize the polar nuclei to form the nutritious endosperm. It is the nutritious endosperm that humans enjoy when they eat seeds. The wall of the ovule develops into a protective seed coat. The wall of the ovary develops into the sweet, fleshy part of a fruit. In this photograph, you can see ovules located within the ovary of a flower. Once the egg within the ovule is fertilized, the ovule develops into a seed. In these photographs of fruits, you're looking at ovules that have developed into seeds located within larger structures known as the fruit. We see seeds within the fruit. We see seeds within the fruit, seeds within the fruit, and seeds within the larger fruit. The ovary wall that surrounds the ovules will, following fertilization, develop into the sweet, fleshy part of a fruit. In these lower photographs, you can see the cucumber flower with its eggs fertilized beginning to develop the cucumber fruit, which encloses the seeds. The first IB syllabus statements of the movie are: draw and label a diagram showing the internal structure of a named seed. All fully developed seeds contain an embryo and some store of food reserves wrapped in a seed coat. The second statement defined germination. Germination is defined as the process by which a plant grows from a seed. The most common example of germination is the sprouting of a seedling from a seed. Here is a simple image of a seed. The diploid embryo, the cotyledon contains the nutrient-rich endosperm, all surrounded by a protective seed coat. Seeds have live cells, the embryo for certain, but the seed is dormant, a stage that serves to protect the embryo from difficult environmental conditions, such as a harsh winter or a dry season. When the environmental conditions are right, the seed germinates. In dicot plants, there are two cotyledons. You should be familiar with this concept from handling peanuts. The two halves of a peanut are cotyledons, dicotyledon. A seed is composed of the diploid embryo, the endosperm contained within the cotyledon, all within a protective seed coat. When the environmental conditions are right, the embryo grows and emerges from the seed. This is germination. The hilum is the location where the ovule was attached to the ovary, a scar that remains from the site of attachment. The micropyle is where the sperm entered the ovule. Here are peanuts. You're familiar with peanuts. You can see the embryos with their embryonic roots and embryonic leaves, and the nutritious endosperm is contained within the cotyledons. It is the cotyledons that people enjoy eating. Here are images of the external and internal structures of a seed. Take some time to review the structures. Can you draw and label a diagram showing the internal structure of a named seed? All of what you see here should be review. One more look. Stop the movie now and spend some time studying this image. Here's the embryo, embryonic leaves, embryonic root and shoot, the protective seed coat, and the cotyledon that holds the nutritious endosperm. The micropyle is where the sperm cell entered the ovule to fertilize the egg. So that brings us to this IB syllabus statement. Design experiments to test hypotheses about factors affecting germination, and those possible factors could be temperature, water availability, light availability, scarification, or the presence of a pollutant. In this graph, you can see the influence of temperature on the percentage of germination. As temperatures increase, percent germination increases until the temperature reaches a critical level above which percent germination decreases. Enzymes denature above certain temperature values. Germination requires water. Without it, seeds will not germinate. The graph displays the change in mass as the seed takes up water. This is called imbibition, and it's measured in time over hours. The photographs display germination, the emergence of the embryo from the seed, following the uptake in water over hours. The uptake of water stimulates enzyme activity within the seed, promoting germination. These graphs display the influence of salty solutions on the percent germination over time. Highly saline conditions reduce germination. Salt would osmotically draw water out of a seed. In other words, when the seeds are exposed to highly saline solutions, germination decreases relative to low saline solutions because the seed is deprived of water. Light could be important to germination for some species. For example, if a tree falls in a forest, soil would be disrupted, providing a flash of light to expose seeds, light that might stimulate germination. The graphs before you display germination over time in varying conditions of light. The solid line curves represent germination in the presence of light, and the dotted line curves represent germination in the absence of light. And germination of N. variegata was highest in the presence of light. A flash of light in the forest would be an adaptive cue as the tree fall opens the canopy for new growth. Seeds that germinated in response to light in these situations would have an advantage over those that did not. Some seeds require abrasion also known as scarification, to germinate. Seeds that roll across sand or open ground would become scratched or scarred. The seed coat would be opened, however slightly. Scarification or abrasion opens the seed to water, but it also ensures that the seed is well dispersed from its mother plant. This graph displays the positive influence of scarification on germination over time. In this photo, we can see germinated Phaseolus species seeds. From this data, we would record 80% germination. So could you design an experiment to investigate factors that influence germination? Using test tubes and seeds of one species, could you isolate one variable while controlling other variables, while ensuring a high trial number, and investigating the influence of water, temperature, or available oxygen on the germination of your chosen species? Summarize the results of the germination experiments shown here. You need to stop the movie now and respond to the prompt. I won't be interpreting the results for you. If you're not sure of the results, see me in class. Here are the results of a germination experiment testing various concentrations of Lipia extract on clover germination. You can see the concentrations of the Lipia extract given here. Lipia is an invasive species introduced to inland river systems in Australia. Lipia releases a chemical that appears to inhibit the germination and growth of other species. And you can see the negative influence of Lipia on germination of clover seeds in these photographs. Here are the results of a germination experiment testing the influence of different sequences of red and far red wavelengths of light on lettuce seed germination. Now, the initial observation was that the germination of lettuce seeds was stimulated by the red light and inhibited by far red light. But the real breakthrough came when experimenters exposed the lettuce seeds to alternating treatments of red and far red light. Nearly 100% of the seeds that received red light as the final treatment germinated. In seeds that received the far red light as the final treatment, germination was strongly inhibited. Here is the setup of an experiment examining the effect of position of seed on the growth of the radicle following germination. Here are supposed results from an experiment testing the influence of a Wi-Fi router on germination. In my view, this experiment needs further investigation. And that brings us to the end of IB Bio Plant Biology Part 8, Plant Reproduction C.